Hello everyone, and welcome to today's webinar, AI-powered AMR detection and surveillance to inform antibiotic precision therapy. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by OpGen. OpGen's portfolio includes services and solutions for outbreak surveillance, infection prevention and control, AMR prediction, and the targeted delivery of antimicrobial therapy. To learn more, visit opgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. And with that, I'd like to now welcome our speaker, Dr. Arne Materna, CEO of Ares Genetics, an OpGen Group company. Dr. Materna, welcome. You may now begin your presentation.
Hello and welcome to today's seminar. My name is Arne Materna. I'm the CEO and Managing Director of Ares Genetics. I have the pleasure today to review together with you the benefits and utility of whole genome sequencing for hospital surveillance and infection prevention and control, and to explore how recent advances in technology can help clinicians adopt this technology into the routine. A few words up front about Ares Genetics. Ares is a subsidiary of OpGen, a company developing RUO and FDA-cleared solutions for molecular infectious disease and AMR testing. Under the Ares brand, our teams develop and commercially offer next-generation genome sequencing and bioinformatics solutions to advance the prevention, the containment and care of patients suffering from healthcare-associated infections. With fully operational NGS service laboratories in Rockville, Maryland, and in Vienna, Austria, we offer genome sequencing-based testing to customers in the US and in Europe, and bioinformatics software globally. But now let's move ahead and review today's agenda. I will start with a brief overview over healthcare-associated infections, or HAI for short. We'll review their impact and the resulting importance of hospital surveillance. In the second section, I will introduce whole genome sequencing, abbreviated WGS throughout this presentation, as a new solution with vastly improved properties when it comes to near real-time outbreak detection and infection prevention and control. We will together review a few recent studies of WGS for hospital surveillance, with a special focus on the patient and the health economic benefits that the various study authors have reported. Concluding, we will discuss barriers for adoption of whole genome sequencing into the clinical routine and present recent technological advances and solutions that lower the adoption barriers and enable hospitals to move forward with implementing WGS. Let's begin with a short overview of healthcare-associated infections, or abbreviated HAI. HAIs are predominantly caused by the so-called escape pathogens, comprised of Enterococcus faecium, Staph aureus, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. Other relevant and increasingly problematic pathogens include, for instance, E. coli, Clostridium difficile, or Candida aureus. What makes these pathogens particularly problematic is that they tend to escape antibiotic therapy by developing and acquiring resistance to common and even last resort antibiotics. Some of the common causes for HAI include central line-associated bloodstream infections, catheter-associated urinary tract infections, surgical site and wound infections, or ventilator-associated pneumonia. HAIs already are among the top 10 causes of death, impacting 1 in 31 hospital patients and 1 in 43 long-term acute care patients. What is also important to know about HAIs is that these infections are preventable through a timely hygiene and infection prevention and control action, which sadly makes HAI the primary cause of preventable death. In the US, about 5% of all hospital admissions result in HAI cases, which are caused, as you can see here in the pie chart on the right, predominantly by surgical site infections, ventilator-associated pneumonia, central line-associated bloodstream infections, C. diff, and catheter-associated urinary tract infections. These cases then impact hospital budgets by increasing the usual length of stay by on average 17.6 days, which is costing US healthcare systems annually something in between 28 to 33 billion US dollars in excess healthcare cost. If you're involved in healthcare or hospital administration, then I'm sure you would immediately find many good alternative uses for these expenditures. But even more important than cost is that the 722,000 registered cases of HAI each year lead to over 90,000 deaths. That is 250 deaths per day in the US alone. And during COVID, this number grew by 15% year on year. In Europe, the numbers look no different, with 7% of all patients in acute healthcare facilities contracting HAI, leading to 35,000 deaths per year. That's another 100 per day, and estimated 16 million additional hospital days each year, causing an excess healthcare cost of roughly 7 billion euros annually. Since HAIs can be prevented by hospital surveillance informing a timely hygiene and infection prevention and control action, these numbers suggest that the current standard approach to hospital surveillance is inadequate. Let's therefore take a closer look on routine hospital surveillance carried out today. Hospital outbreak detection, investigation, and control follows a paradigm that essentially in many places is unchanged from the methodologies described by John Snow, who in the mid-1800s conducted a series of investigations of cholera outbreaks in London, both to discover the cause of the disease and to prevent its recurrence. Snow relied for his studies on observations of disease in a geographic and a temporal context to infer outbreak activity and disrupt the source of the disease, which for cholera were some of London's water sources. In today's hospitals, daily surveillance is undertaken for patients who are positive for pathogens of particular importance. The detection of 2 or more patients who are positive for the same indicator organism is investigated using hospital databases, ward visits, etc., in order to determine whether cases have overlapped in time and place, and to identify any other factor that could link them. All of the available information is then reviewed manually to assess the probability of an outbreak and the need for either further investigation, for example, the screening of other patients, staff, or equipment, or for interventions, for example, through enhanced infection and control measures, hygiene routines, or cleaning. The problem with this approach is that factors such as time and location can be circumstantial, leading to cases when healthcare-associated infections, which require urgent infection prevention and control action, and cases of community-acquired infections, which do not require any action, cannot be told apart. This is where methods for pathogen typing come into play. These methods establish genomic relatedness between individual pathogens that were isolated from patients. Related isolates are a much stronger indicator of the observation of a new or active outbreak cluster. The issue with most typing methods, however, is twofold. Genotyping can be slow, especially when samples have to be first collected and sent to partner labs or research collaborators. Also, most methods, such as pulse field gel electrophoresis, SPA typing, or MLST, which are all widely used, consider only a very limited number of genomic markers or locations. Therefore, these methods suffer from a poor resolution for isolating and discriminating outbreak clusters and transmission chains in the data. And to be clear, data that come 2 or more weeks after collection and that cannot resolve outbreak clusters well do lose their value for guiding infection prevention and control action. Here, whole genome sequencing essentially represents a paradigm shift in hospital surveillance. Via this approach, each case of a surveillance critical pathogen is submitted to sequencing, which is the only method that offers a type of relatedness analysis that is scrutinizing and comparing the entire genome of each isolate with single nucleotide level resolution. As a result, low-resolution typing data and any subjective guesswork on relatedness is essentially replaced with an objective measurement of relatedness or distance between current and past cases. The advantage over traditional or even molecular typing methods hereby is that the process of detecting cases belonging to new or already active outbreak clusters can essentially be automated. And in addition, whole genome sequencing provides highly resolved transmission chains, which make the identification of effective hygiene and infection control interventions much more straightforward. Whole genome sequencing is thereby not less, but rather more sensitive or specific than other methods. What has always intrigued us about whole genome sequencing, and the reason why the ARES team at OpGen is fully focused on sequencing as a core technology, is that the method can provide accurate information on pathogen ID, antibiotic resistance, genomic relatedness, outbreak clustering, and pathogen transmission, all in a single assay. And thanks to the continuous innovation of NGS platform technologies, both cost and turnaround times have substantially decreased. With as little as 4 to 6 hours turnaround time, clinical microbiology labs with advanced NGS and bioinformatic capabilities already in-house can in theory today enable same or next day reporting. Here an example of a process for whole genome sequencing based outbreak detection as implemented into healthcare settings at the University of Pittsburgh Medical Center by a team of researchers led by Dr. Alexander Sunderman. In this model, every clinical culture from a patient infected, for instance, with Acinetobacter species, Pseudomonas species, extended spectrum beta-lactamase producing Escherichia coli, Klebsiella, and a range of other indicator organisms were submitted to whole genome sequencing if certain inclusion criteria for hospital admission and observation dates were met. If whole genome sequencing then revealed a new or active outbreak cluster for any of the patient isolates, and if the transmission route could be bioinformatically established, the infection prevention and control, or abbreviated IPC teams, were notified immediately. If transmission routes could not be resolved, for example due to the number of cases still being too sparse, the epidemiologists would first review the relatedness patterns in the context of the information in the medical record before reporting to the IPC team. It is of note that a substantial part of the interpretative WGS or whole genome sequencing process can be automated or at least guided by bioinformatics software. Critical hereby is that the software has to be accurate and ideally validated, easy to use, that it offers rapid turnaround times regardless of sample volumes, and that the software ensures that the data are both safe and securely backed up, that it acts in compliance with data privacy regulations, such as the HIPAA Act, and last but certainly not least, that the information is condensed into a format that is conducive to action by IPC experts. I'm now switching gears as we will review a number of recent studies to assess the patient and economic benefits, in other words, the clinical utility, of implementing whole genome sequencing for routine hospital surveillance. I'd like to start with a study conducted by the aforementioned team in Pittsburgh, led by Dr. Alexander Sunderman. In a study published in 2020 by Kumar and co-workers, the authors retrospectively analyzed whole genome sequencing data gathered between 2011 and 2016 from 11 HAI outbreaks involving 89 patients. The researchers bioinformatically assessed relatedness and established transmission networks, and then used these data to model the impact that whole genome sequencing would have on disrupting hospital transmissions and on reducing mortality if routinely applied. The authors' predictions are shown in the table on the right-hand side of the slide and led to the conclusion that transmissions could be reduced by more than 45.7% and mortalities by 51.7%. Interestingly, the cost of whole genome sequencing projected by the model were outweighed by the savings due to the reduced hospitalizations and excess healthcare cost, leading to an overall saving rather than a cost. Probably encouraged by these predictions, Dr. Sunderman and his team further refined their process for whole genome sequencing-based outbreak detection and embarked on a study published in 2021, which retrospectively analyzed whole genome sequencing data and associated medical records for 2,752 patients. The team then compared the outputs of their whole genome sequencing process to the standard of care at the local hospital to project health and economic benefits of whole genome sequencing. The observations, again shown on the right, confirmed their earlier findings. First, the diagnostic yield for detecting outbreaks and as well as implicated patients was drastically increased through WGS. Even when considering that the standard of care results excluded colonizations, which makes this a bit of an apples to oranges comparison, it can be safely assumed that the standard of care misses many incidents of HAI outbreaks. Overall, the study predicted a reduction in transmissions, associated 30-day readmissions, and mortality. Remarkably, and driven by the reduced hospitalization healthcare costs due to HAI cases, the net savings per detected transmission event were estimated to be as high as $10,900 per case. The authors also communicated that a turnaround time of roughly 2 weeks appeared to be sufficient for the local IPC teams to sustain a reduction in transmission events at the clinic. The projected economics, here shown by the upper and lower estimates for net savings depending on the number of detected transmission events, impressively demonstrated that whole genome sequencing, regardless of the number of cases, makes sense purely from a cost perspective. An argument in favor of whole genome sequencing that is made even stronger when considering the patient benefit and the method's ability to reliably identify outbreak clusters, here shown in the center of the circular dendrogram, and to effectively discriminate them from the periphery of community-acquired infections, and to do so universally across critical pathogens. For the third study, we shift our focus to Australia and to a group that has expanded the focus of their investigation from the local hospital to the regional healthcare level. The research teams of David Patterson and Patrick Harris at the University of Queensland in Brisbane conducted a thorough analysis of the budget impact of routine whole genome sequencing-based surveillance of 6 critical multidrug resistant pathogens at 27 hospitals across the Australian state of Queensland. In order to generate a solid data foundation for their analysis, the teams conducted prospective whole genome sequencing outbreak analysis for 2 years and collaborated with a team of healthcare economists familiar with the local cost of care. Before we dive into the details, a little spoiler up front. The study published by first author Louisa Gordon in 2021 concluded that whole genome sequencing surveillance leads to a statewide reduction of HAI-associated death of 32%, a reduction in colonized patients of 38%, and a net reduction of HAI-associated healthcare costs of 18%. The findings we will now review have compelled the state government to support the implementation of routine whole genome sequencing to the tune of 25 million Australian dollars. As I already mentioned, the team collected data from 27 hospitals and considered the entire state of Queensland, the second largest and third most populous in Australia, for their analysis. The costs were modeled based on costs for hospitalizations, sequencing, and epidemiology action. The assumption on HAI incidence considered the annual hospitalization rates, the point prevalence for HAI in Australia, which is just shy of 10%, and the multidrug resistance rates and their trends. The costs were then aggregated for the state of Queensland based on the expected number of multidrug resistant isolates arising in Queensland hospital patients given the point prevalence. The surveillance data for this study came from a 2-year research demonstration project of prospective whole genome sequencing for isolates of suspected outbreaks. The interventions compared by the authors were whole genome sequencing versus the standard of care for MDR pathogens. An implementation of whole genome sequencing was assumed for 6 common multidrug resistant organisms, including methicillin resistant Staph aureus, extended spectrum beta-lactamase producing E. coli, vancomycin-resistant Enterococcus faecium, ESBL-producing Klebsiella pneumoniae, carbapenemase-producing Enterobacterialis, and carbapenem-resistant Acinetobacter baumannii. The study compared the following primary and secondary outcomes for both interventions: expected hospital costs, counts for patient infections and colonization, and death from bloodstream infections. The authors have actually calculated cost annually across 5 years from the base year 2020 onward. But for simplicity, I only highlight here the impact of year 1 of whole genome sequencing. The study findings projected that in year 1, when only relying on standard of care testing, 97,539 patients in Queensland were expected to be infected or colonized with one of the 6 multi-drug resistant pathogens. A timely infection control measure as informed by whole genome sequencing surveillance could avoid 36,726 infected or colonized patients and avoid 650 deaths. These numbers, which are highlighted here in green on the right-hand side table, deliver, I think, a very clear message about the patient benefit that can be realized through routine whole genome sequencing surveillance. Moving on from the patient benefit to the economics, let's take a look at the impact on healthcare cost next. The total cost under the standard of care was 170.8 million Australian dollars in 2021. Whole genome sequencing surveillance costs an additional 26.8 million Australian dollars, which however was offset by reduced costs for cleaning, nursing, personal protective equipment, of course also shorter hospital stays and reduced antimicrobial treatments, leading to an overall cost saving from whole genome sequencing surveillance of over 30.9 million Australian dollars in year 1 alone. In summary, the authors established that whole genome sequencing surveillance provides a clear benefit to patients as well as to health economics. To demonstrate that these findings are rather reliable, the authors also conducted a sensitivity analysis which showed that these cost savings remained when input values were varied at 95% confidence limits. The last study I'd like to present was published almost exactly 1 year ago and conducted by Dr. Norelle Sherry and co-workers at the University of Melbourne in the Australian state of Victoria. Also here, the authors investigated in a prospective multi-center study the implementation of whole genome sequencing surveillance regionally across 8 hospitals. To do so, 2,275 isolates were collected from 1,970 patients and sequenced. And again, the focus was on HAI pathogens, in particular on vancomycin-resistant Enterococcus faecium, extended spectrum beta-lactamase-producing E. coli, and Klebsiella pneumoniae. Out of these 2,275 isolates, 844 were implicated by whole genome sequencing as genetically related. 354 of these cases were likely to be caused by healthcare-associated infections, with all the consequences to patient well-being and healthcare cost we have just learned about. Another 253 of these cases were possibly linked to HAI, and only 263 were unlikely to be caused by healthcare-associated infections. This data set again underlines the pervasiveness of healthcare-associated infections and the shortcomings of the current standard of care for hospital surveillance across hospitals and networks, also in highly developed areas of the world. Allow me to be blunt at this point, but anyone believing that their hospital is not suffering from HAIs simply suggests that they are not looking, at the expense of patients and payers. What I do find interesting about the Melbourne study in particular is the perspective on whole genome sequencing from an epidemiologist's point of view. The team found, for instance, many unexpected transmissions warranting interventions that were specific to only certain wards. They also emphasized the distinct advantage of whole genome sequencing in detecting HAI clusters and transmissions that would have been complicated to identify with traditional epidemiological analyses. And they conversely stressed the cost and stewardship advantage of reducing IPC interventions, where whole genome sequencing could confirm that cases were unrelated and caused by community-acquired as opposed to healthcare-associated infections. Learnings at the regional level included that incidence of related VRE transmissions were higher in some hospitals than in others, justifying hospital-specific action or changes in hygiene. Also, the team found that MDRO infections that were previously considered community-acquired were actually related, providing evidence for previously unrecognized HAI outbreaks. Finally, the fact that 86% of HAIs were caused by VREs was able to inform changes in public health policies at the state level. Another aspect that is often overlooked and that the authors of this study explored further is how the rather rich information provided by genomic relatedness analyses in the form of dendrograms, for instance, can be condensed into reports that IPC teams can act upon. In their example, they share with Infection Prevention and Control, or IPC, as little as an alphanumeric sequence type for each isolate, and a color-coded traffic light indicator to classify genomic relatedness into high, suggesting likely transmission between the listed patients, medium, potentially warranting further manual investigation under consideration of the patient record, and likely unrelated, essentially advising against IPC action. So summarizing so far, HAIs are the primary cause of preventable death among hospitalized patients. Whole genome sequencing surveillance of isolates can confirm or refute epidemiologically linked infectious pathogens, thereby discover new outbreaks or incidents of active known outbreaks. In the same assay, whole genome sequencing can accurately confirm pathogen ID, discriminate between different strains, classify novel pathogens, and more. Whole genome sequencing surveillance, when adopted into the clinical routine, saves lives and has the potential to reduce HAI incidence by up to 70%. Finally, the cost-effectiveness of whole genome sequencing for HAI surveillance has been demonstrated at the local and regional level in robust, independent, retrospective, and prospective multicentric studies. If we consider for a moment the definition of clinical utility of an intervention, which is defined as the patient benefit divided by the total cost of care, then these 4 studies we just reviewed all suggest a high clinical utility of whole genome sequencing and establish a favorable value proposition for adopting pathogen whole genome sequencing into routine clinical practice for surveillance. Which brings me to the final section of this seminar, in which we discuss technological advances and solutions that help healthcare providers accelerate the adoption and establishment of whole genome sequencing surveillance. Let's review some of the challenges that can complicate the adoption. One barrier is technology access, hampered by a lack of local capacity for whole genome sequencing. To build local capacity, clinical microbiology labs need to expend the capital equipment cost to bring NGS platform technology in-house and to bring on board trained staff, which is something that has been in short supply since COVID-19. Even with technology and staff on site, establishing and validating laboratory-developed tests is at least initially a burden. Another very important factor is turnaround time. Whole genome sequencing surveillance workflows have to be able to inform IPC action within no more than 2 weeks after sample collection, and ideally faster. Especially when first adopting whole genome sequencing surveillance and when the number of samples per run can still be low, running NGS platforms below capacity to meet turnaround time requirements can again result in higher per sample costs. It goes without saying that any clinical test or intervention has of course to be validated and reliably offer accurate performance, which for whole genome sequencing is not only a matter of the wet lab workflow, but especially is especially of importance for bioinformatics and data interpretation and actionable reporting. Setting up and maintaining bioinformatics in a way that fits into a CLIA environment traditionally requires specialized personnel and access to scalable compute hardware, which is driving up the total cost of ownership continuously generated by the need for bioinformatics. Depending on bioinformatics, of course, also comes with the burden of ensuring that any tools that are used comply with data privacy protection and patient confidentiality standards such as HIPAA. Our R&D and Aries product development efforts at OpGen therefore focus on lowering these barriers to adoption. In a nutshell, we enable enhanced surveillance for detection of microbial outbreaks in healthcare facilities by providing validated whole genome sequencing services and comprehensive genomic analyses that enable earlier and more sensitive identification of outbreaks in order to prevent in-facility transmission sooner and reduce HAI, AMR, and their impact on patients in healthcare. We distinguish between full service and analysis only. Full service for isolate sequencing, dubbed ARIES ISS, includes sample processing and whole genome sequencing using workflows that we have fully validated following CLIA guidelines for both short and long-read sequencing platforms. Our process is streamlined to offer short turnaround times of as little as 5 business days in certain geographies. And to facilitate sample collection, we provide kits along with detailed instructions and everything required for safe collection, storage, and express shipping of clinical isolates to our facilities. We thereby recognize that sample collection procedures can differ from hospital to hospital. For example, some use slants instead of plates for storage and shipping. And our teams are fully prepared to customize sample collection to best fit your process. Full service also includes data analysis, including pathogen ID and typing, outbreak cluster detection, as well as comprehensive genomic characterization of AMR plasmids and virulence markers. Unique to ARIES technology, and another benefit we believe routine clinical whole genome sequencing can enable, is the machine learning power prediction of phenotypic susceptibility and resistance to antibiotics directly from genomic data. Such antibiograms, generated through this proprietary process we refer to as genomic AST, are therefore routinely incorporated into our isolate reports. All data, results, and reports, in addition to interactive tools for outbreak and transmission analysis, are accessible via the ARIES Cloud web application. And just like for sample collection, our team will work with your IPC and epidemiology specialists to customize the reporting and ensure a seamless integration into your clinical workflows for data processing. The analysis-only option is delivered via the web application ARIES Cloud. ARIES Cloud is an easy-to-use, fully featured standalone software that is designed to supply clinical microbiology labs with in-house NGS capacity, with validated, fast, scalable, and secure analytics, and with all the data interpretation and reporting features that full-service customers enjoy. This slide provides an overview over the different ARIES services that OpGen service labs are currently offering, including ARIES ISS, the service we just described for rapid isolate sequencing and the focus of today's presentation, but we also offer ARIES ISP, a comprehensive target enrichment panel for environmental AMR surveillance, which screens for over 14,000 AMR markers included in the ARIESDB database, and ARIES ID, an amplicon sequencing service for the culture-free identification of pathogens with species-level resolution directly from patient specimen. To fully leverage the potential for rapid turnaround times of real-time long-read sequencing using nanopore technology, we have validated ARIES ISS not only on the current de facto standard Illumina, but also on sequencing devices provided by Oxford Nanopore Technologies. These platforms have properties that make them ideal for infectious disease applications. Sequencing on ONT is performed by translocating long DNA strands through biological pores and across a membrane. Different electrochemical properties on either side of the membrane lead thereby to a measurable electrical current through the pore. And this current changes depending on the sequence context as DNA single strands pass through the pore, creating thereby electrical signals that can be converted into a sequencing readout. Advantages of this technology include, for instance, that sequencing is performed in real time on platforms that do not require any complicated onboard liquid handling or optical sensing, which significantly reduces platform costs and importantly also NGS runtimes, down to as little as a few hours. Like all ARIES sequencing services, also ARIES ISS has been fully validated following CLIA guidelines. And to help clinicians adopt whole genome sequencing into their local labs, we determine diagnostic performance of our assays, typically in multicentric studies, and we publish not only the performance results, but also the validation protocols, including the reference materials we used. Shown here on this slide are the validation results for isolate whole genome sequencing via ARIES ISS and ARIES Cloud And as you can see, pathogens were identified and typed as required for outbreak analysis with the highest sensitivity, specificity, and reproducibility. Ares Cloud has been designed to facilitate rapid whole genome sequencing run data interpretation in under 1 hour through a software that scales to any sample throughput at low and importantly at predictable cost to the hospital or the laboratory. Data analysis after data upload is fully automated and requires zero user intervention. And Ares Cloud provides actionable reports and easy-to-use outbreak analysis features for relatedness and transmission analysis in the context of EHR metadata. All bioinformatics pipelines have been fully validated and allow clinical microbiologists to comprehensively type and characterize pathogens, predict AMR, and to detect and explore outbreak clusters and transmission events at the highest possible resolution. Before I conclude the presentation, I would like to digress for a couple of minutes and invite you to join me to an excursion into capabilities beyond outbreak detection that whole genome sequencing can offer when coupled with AI and predictive modeling. Core to the analytical capabilities that Ares teams have been developing since 2017 is a microbial database we refer to as AresDB. AresDB hosts over 130,000 carefully selected and curated clinical pathogen genomes. And what makes this database stand apart from other genome databases is that for each isolate genome, also the associated phenotypic antibiotic susceptibility testing or AST measurements have been obtained under controlled lab conditions. The resulting data sets of associated genome on one hand and AMR phenotypic information on the other hand can be mined for previously unknown biomarkers for AMR, leading to a comprehensive selection of well-annotated genes and mutations associated with antibiotic resistance. But the main purpose of these data sets is to serve as training data for machine learning models to predict antibiotic susceptibility, or in other words, how a given microbe would behave when exposed to different antibiotic compounds. Unlike rule-based approaches, as used by ResFinder or AMR Finder, our AI models do not depend on simple presence-absence of known genetic markers alone, but allow for polyvariate sequence signatures, including, for instance, in non-coding regions that may affect gene expression. The machine learning approaches we have chosen and optimized over the years rely on a process referred to as stacked ensemble modeling. Instead of only learning sequence signatures, like k-mers, that are predictive of phenotypic AMR, these algorithms can associate AMR phenotypic data with a whole stack of sequence and sequence-derived information, such as amino acid information, gene and genome structure and synteny, or mutation impact scores. Considering also information that is more robust to sequencing errors makes these models essentially out-of-the-box highly accurate for different NGS data types, which is one of the reasons why we see highest accuracy across all sequencing technologies we have evaluated and validated, which include Illumina, Oxford Nanopore, as well as PacBio. We've shown in a recently published paper that these models can, with only minor modifications, even be applied to metagenomic sequence data and still predict susceptibility with a balanced accuracy of 90% and with major and very major error rates superior to those of rule-based approaches. The performance of these genomic AST models has not only been evaluated by us in-house, but also by our clinical collaborators in numerous studies. Since the original multicentric clinical study, which has been assessing the performance of Ares genomic AST in the context of isolates collected from respiratory tract infection patients, highlighted here in red on the left-hand side of the graph, several collaborators have explored a number of complex and edge cases. For instance, studies performed at Mayo explored genomic AST in E. coli bloodstream infections and Acinetobacter baumannii infections. Or a study conducted at Johns Hopkins investigated genomic AST model performance in carbapenem-resistant Enterobacteriaceae, which however were negative for the resistance-conferring carbapenemase gene, and therefore represent a real challenge for any molecular or genomic approach to AMR testing. And another study conducted at Mayo assessed genomic AST to perform also favorably compared to other methods in multi-drug resistant Pseudomonas aeruginosa isolates. Then finally on the right and highlighted in green are the results of another internal study that's been demonstrating the improvements in genomic AST our teams were able to make in a time window of about 18 months over the original multicentric study on the very left. And finally, before I end this excursion into genomic AST and conclude the seminar, here a slide that shows how selective curation of isolated genome and phenotypic AST data to train AI models can contribute to an improved model performance. The x-axis shows the inclusion of additional data sets, which we obtain, for instance, through collaboration with reference laboratories or public health laboratories. The graph is showing how the inclusion of these additional data sets for certain pathogen drug combinations is then leading on the y-axis to a reduction of here very major error rates below the regulatory threshold for AST-IVD devices of 1.5%. But now, switching back to the original topic and wrapping up this seminar on whole genome sequencing surveillance. And in conclusion, we have established that whole genome sequencing surveillance in healthcare settings is of high clinical utility. Again, it can confirm or refute epidemiologically linked pathogens and rapidly as well as accurately detect HAI outbreak clusters and transmission events, which in turn can guide improved IPC action. Current barriers to adoption come in the form of high upfront equipment cost, the complex and specialized wet lab and bioinformatics workflows required for whole genome sequencing surveillance, and the lack of staff trained in these workflows. Lack of access to complex yet robust and scalable bioinformatics, and of course, in the form of the challenge that's inherent to communicating complex genomic information in a format that is actionable from an IPC standpoint. At OpGen, we leverage and contribute to technological advances in order to lower these adoption barriers. With Ares ISS, we offer a rapid turnaround time send-out to Ares labs, allowing hospitals to defer the initial upfront cost involved in adopting whole genome sequencing surveillance. To streamline the sample collection and shipping through the microbiology lab, we offer fully return-labeled collection kits and protocols for critical bacterial and fungal pathogens. End-to-end workflows for whole genome sequencing, as well as the standalone bioinformatics software Ares Cloud, are fully validated and provide access to easy-to-use data interpretation and actionable reporting. Our team will work with your microbiology lab and IPC teams to customize sample collection procedures, shipping, and IPC reporting to best meet your local requirements. All data are safely and securely stored and backed up, and Ares Cloud is ready to act as your own data interpretation and reporting platform as soon as your hospital is ready to bring whole genome sequencing in-house. So in summary, we are here to work with you from developing the business case for whole genome sequencing to establishing its utility at your hospital and all the way to building full and future-proof on-site capacity. And with that, I would like to thank you very much for your attention and invite your feedback and questions.
Thank you, Dr. Materna, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. We've got some great questions already coming in from our audience here. The first question I'll start with, Dr. Materna asks, or mentions, I am excited to hear that Ares Genetics is publishing validation protocols for the isolate sequencing workflow to support other labs and their adoption of NGS. We are planning to establish NGS capabilities in our lab next year. Do you offer some kind of service that would help labs with the setup of this or similar workflows?
That's an interesting question. Can you hear me still loud and clear?
Yes, we can.
Excellent. Good. Yes. So we are, of course, happy to share studies and protocols and also the reference materials we use for validating our assays. And we have, our team on standby to elaborate on these. However, I need to point out that to be compliant with regulations, the actual validations of laboratory developed tests, or LDTs, need to be independently carried out by the clinical or reference lab that is seeking to implement them. And in fact, to lighten that particular burden for labs with independent access to NGS was one of our main motivations for publishing and sharing our validation protocols, as well as results and reference materials. And also for supplying Ares Cloud to enable these labs to independently generate pathogen data with the same level of accuracy and the same reporting and data interpretation features that our service labs in Rockville and in Vienna enjoy.
Great, thank you. Another question I have for you here mentions and asks, we have sequencing capabilities in-house for 16S, and we consider using the platform also for surveillance. How fast is Ares Cloud, and what price models for Ares Cloud do you offer?
Okay, Ares Cloud scales with the sample throughput. This means regardless of whether you analyze 1 or 100 isolate whole genome data sets, the software scales up and down compute resources on the cloud as needed to make sure that you obtain results in under 1 hour. You can buy Ares Cloud via a simple subscription model, and the price for a 1-year subscription is fixed, so you don't encounter any surprises. The subscription price thereby depends on the size and the anticipated surveillance load of your institution. And it covers full use of the software, including, for example, sequence data analysis, reporting, the continuous interpretation, for example, of outbreak data, as well as compliance, safe, and secure data storage and backup. For detailed questions, for example, on entry-level pricing, software trials, etc., I suggest to get in touch with us via the email here on the slide, or via the contact details provided on our website.
Thank you, Dr. Materna. Another question I have for you here. We are currently building a business case together with the hospital administration for the adoption of WGS. Are there any suggestions or lessons learned from your customers that you can share with us?
That's a very good question. In fact, and honestly, with a bit of hindsight, I would assume that most decision makers actually underestimate, on one hand, the amount of outbreak activity that is healthcare typical in their institution, and on the other hand, the cost impact that these HAI outbreaks have on their budgets. So a probably good way to assess this impact is by conducting a pilot over time, and choose a time frame and a sample volume that is representative. I would actually strongly recommend to conduct such a pilot in a prospective manner, as this allows you to readily assess also your procedures for sample collection, shipping, as well as the communication with your IPC teams in a real world as opposed to a theoretical setting. I think I would further recommend to focus on a single surveillance relevant pathogen species for this pilot. And I'm sure your IPC team can tell you which species is likely to cause the most HAI outbreaks at your hospital. The advantage of focusing on a single species is that you will collect a higher number of comparable data points and thereby increase the statistical power of your predictions on patient as well as economic impact and benefits from whole genome sequencing surveillance. So with a single pilot, which you may finance, for example, as a research project, and with sequencing as little as, say, 100 isolates, you could generate sufficient data to inform a cost-benefit analysis and then extrapolate the results across other HAI species.
Great, thank you. Now, this next question has a few separate parts to it, and I'll go ahead and start here. When comparing similarity or divergence of 2 whole genomes, how do you set the cutoff or tolerance for genetic variation? Is one nucleotide difference across the whole genome sufficient to conclude not related, or are more differences required to conclude not related? And how do you consider the different speeds of evolutionary clocks in different regions of the genomes?
This is a great question and very technical, focusing on the analytics for cluster detection, I believe. So While in local outbreak settings or for some species, SNP thresholds might be sufficient. This approach, and this is, I think, what the question is addressing, this approach can indeed be imperfect in certain situations. And I think especially in large multi-year retrospective analyses, like for instance, big public health studies. There are, however, methods emerging, like for instance, probabilistic approaches. Such approaches, they still consider genetic distance, but also sample time, together with certain models for SNP acquisition over time and transmission events over time. And then to base putative transmission clusters on the probability that the cases are separated by a certain minimum number of transmission events, for example. Certainly choosing a sufficient approach for any given species and setting can be important. And also the data you gather over time can inform the appropriate approach.
Great. Now, I did see a question hop in here in regards to obtaining the CE credits. So I just want to reiterate that you can find those continuing education credits in that continuing education window that's located on the bottom of your screen. If you click there, you will be prompted to obtain those credits. All right, and we have time for a few more questions here. So I'll ask this next one. Are the sequences contributing most to resistance prediction associated with known AMR elements, or are they lineage and or plasmid markers or something else?
No, I think it's fair to say that the sequences and KMRs that most contribute to resistance prediction are not necessarily always linked to known antibiotic resistance genes. Of course, the well-established known genes that lead to antibiotic resistance, such as certain transporters or, for example, beta-lactamase inhibiting genes or carbapenemases, etc., all can sorry, let me catch my train of thoughts. So essentially the presence of certain well-established resistance genes is a strong predictor in itself. And if these predictors or genes are present, they also are contributing to the KMR profiles that the model pick up on for resistance prediction. However, there are many other factors, genetic factors, and often these genetic factors can be polyvariate. For instance, multiple genetic factors can occur simultaneously in different locations, for instance, in a manner that might affect gene expression, if these factors are, for example, located in upstream regulatory regions. And considering not only presence, absence of such factors, but their combinatory effect is important. As if you think about this from a essentially ecological perspective, then any mutation that you acquire can contribute synergistically or antagonistically to an organism's fitness, and this has to be this all can be factored in by predictive models, which is something that rule-based approaches cannot do. And so, as a consequence, I don't think it is surprising that such polyvariate genetic factors can be very important, especially in cases where certain known resistance genes, for example, the CP negative case that I explained in the presentation, where they are absent. So I think it cannot be said that these are always associated with AMR markers, but I think it's a fairly good mix of well-known AMR genes as well as polyvariate genomic factors. Of course, also point mutations and such. I hope that answered the question. If not, please feel free to reach out to us and we can go into this in more detail.
Great. Well, thank you, Dr. Materna. We are going to go ahead and wrap up here and conclude our Q&A session today. So I want to thank you again, Dr. Materna, for your time today and your important research. We would also like to thank LabRoots and our sponsor for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you had provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.